To get started. So we have signal up there, everything's working. All right, so where are we headed? We're going to finish that story from yesterday. We're going to start with a couple of slides. But fundamentally, what we're going to be talking about today are the G protein coupled receptors and their effects on the heart. Much of this will be familiar to you. These are the G protein coupled receptors we introduced when we discussed the endocrine system. And it's the same, pretty much the same story. We're going to short circuit it a little bit because we're going to see, particularly in the pacemaker cells, the main operating character will be the cyclic AMP. So what we're going to be doing is we're going to be binding norepinephrine to those beta-1 receptors. The beta-1 receptors now will, through the G protein coupled receptor system, will now increase the amount of cyclic AMP. With increased amounts of cyclic AMP, there's a higher probability of opening those HCN channels, which makes the rate of rise of the ramp potential that much faster. So we can regulate heart rate much largely through the sympathetic and parasympathetic systems. And we'll see that we'll talk first about the parasympathetic system. And we'll do this mostly because the parasympathetics innervate only the pacemaker cells. So we're not going to have to worry about innervation of the contractile cells with the parasympathetics. We're going to find that there's a G protein coupled receptor here. This G protein coupled receptor has a name. It's called a muscarinic receptor. It's this particular one. There are 4 muscarinic receptors. Some places you'll see 5 of them. And this is M2. What M2 does is M2 is a subtype. It's a type of G protein coupled receptor. It's a particular type of G protein coupled receptor that binds acetylcholine. And in this case, it's going to bind acetylcholine released from the postganglionic parasympathetic neuron. That acetylcholine binding to the G protein coupled receptor will activate one of those alpha subunits we learned about some time ago called G-alpha I. Remember, I stands for inhibitory. So what I is going to do is it's going to reduce the activity of adenylyl cyclase. By reducing the activity of adenylyl cyclase, we have now downregulated the concentration of cyclic AMP in the cell. And in so doing, we have less cyclic A available to open those HCN channels, so we're less likely to get sodium influx. So the rate of rise of that ramp potential leading to threshold in those pacemaker cells is much slower. It takes longer to get threshold. So now we have a slower heart rate. So you can see how this is going to be rest and digest, slow down the heart rate. If we can turn that story on its head, we can think about the sympathetic branch. And in the sympathetic branch, we'll see that the norepinephrine is released from the postganglionic sympathetic neurons. That norepinephrine binds to G protein coupled receptor. It's often referred to as an adrenergic receptor, but it's a particular kind of adrenergic receptor. We're going to see there are several subtypes of these adrenergic receptors, the G protein coupled receptors that bind norepinephrine. The one for today is going to be beta-1. Beta-1 is found in the heart. It's found both on the conductile cells and the contractile cells. So we're going to have to think about how the beta-1 receptors are affecting both the rate and rhythm and the strength of contraction. So we'll walk our way through that. The first thing that's going to happen, I think you can see from the other side of the story for the M2 receptors, is that we're going to use the G-alpha S to increase activity of adenylyl cyclase. By increasing adenylyl cyclase activity, we've now increased the concentration of cyclic AMP. Remember the HCN channels now. They have 4 binding sites for cyclic AMP. The higher the concentration of cyclic AMP in the cytoplasm, the higher the probability that you'll bind several of those sites, several of those 4 sites. And the more of those sites that are occupied, the greater the probability that channel swings open. As the channel swings open and lets sodium in, it depolarizes the membrane potential of the pacemaker cells. The faster we're letting sodium in, the faster we're depolarizing, and the faster we're going to generate the action potentials in these pacemaker cells and thereby increase the heart rate. So we'll walk our way through that. But we'll see also that the norepinephrine, should be nor, the beta-1 receptors for norepinephrine in the sympathetic branch are also going to be found on the contractile cells. So where the parasympathetic innervation is largely restricted to the pacemaker cells. Now we have the sympathetic innervation innervating both the contractile cells and, the pacemaker cells. So in the pacemaker cells, the effect of the norepinephrine is almost entirely to increase the cyclic AMP. But when we start talking about the contractile cells, we now go back to the older story that we had before, where the effects of G-alpha-S are not only to increase adenylyl cyclase activity, increase cyclic AMP, but the cyclic AMP then increases phosphokinase A. And so now we're going to start phosphorylating these various pumps and channels. And so by phosphorylating the calcium channels, we're going to increase the amount of calcium. By increasing the amount of calcium in these contractile cells, we're going to increase the strength of contraction. But now recognize what we're doing. It's a kind of twofold event here. In one case, we're increasing the heart rate, but now we're also increasing the amount of calcium in the cytoplasm. How do we stop? How does the heartbeat come to an end? The contraction comes to an end when the calcium comes off the troponin, C. So now we have a double whammy here. We've increased the amount of concentration, we've increased the concentration of calcium in the cytoplasm, which now also increases the strength of contraction. We've also increased the rate. So the other thing that's going to be phosphorylated here is we're going to phosphorylate the pumps. The calcium pumps are now going to be phosphorylated to increase the rate of movement of calcium from the cytoplasm back into the endoplasmic, or the sarcoplasmic reticulum. And so we'll walk our way through that. Then we'll see there's a real effect in the cardiac cycle. We'll see that unlike Starling's law, where we increase, the end diastolic volume, in the case of increased contractility, we'll actually reduce the end systolic volume. So we'll get a greater stroke volume simply by squeezing more blood, being more efficient about the heartbeat. And we'll talk about that. Then we're going to talk about systemic vasculature. Now this story we can go through pretty quickly because we've covered this in some detail when we were discussing the details of the coronary vasculature. And the story is pretty much the same here. We're going to have arteries, arterioles, capillaries, venules, veins, vena cava, right atrium, same circuit. It's going to be a closed circuit. The fluid is never going to leave those tubes. And we have a series of tubes having various functions. The arteries, as we know, and arterioles are going to be carrying blood away from the heart. The capillaries are going to be the exchange vessels. That's where we're going to exchange oxygen and nutrients. The blood will flow through the capillaries, back through the venules, veins, and vena cava. So once we get to the venules, we're now in the return circuit. We're coming back to the heart. Again, the terminology. Veins are carrying blood toward the heart. So we'll carry it through the system back to the right atrium. But this system is also under autonomic control. Here the autonomic system, and I'm going to simplify it a lot, but in the autonomic system here is under mostly the control of epinephrine and norepinephrine, which bind to a particular kind of adrenergic receptor called an alpha-1 receptor. The alpha-1 receptors are on smooth muscle. And remember yesterday we said the three possible targets are cardiac, smooth, and glands. Well, this will be on smooth muscle. This will be on the smooth muscle of the vasculature. The function of the adrenergic innervation is to activate a G protein coupled receptor, the alpha-1 receptor. That leads to smooth muscle contraction. So from Poiseuille's law, when we talked about the coronary vasculature, you can see what's going to happen here. We're going to reduce the flow through those particular tubes. But here we have so many different tubes through which we can send fluid. By closing down one with using smooth muscle, we can allow that flow to go to other places. This is basically what's happening when you go outside on a really cold day. And when you go outside on a really cold day, if you'll notice sometimes your hands get white, what you're doing in order to control and maintain core temperature is you're using this smooth muscle to close down arterioles leading to your skin to reduce blood flow. And that shunts more of the blood back into the core so you retain heat. So we're using these things at all times. But in this particular case, we're going to talk about the effect of alpha-1 adrenergics on smooth muscle contraction. And then we'll finish that story with the dissipation of energy along the wall. Again, this is just a restatement of Poiseuille's law. And what it's going to tell us is that almost all of the friction between the fluid being pumped through these vessels, almost all the friction between the fluid and the walls is within a very, very narrow gap, a narrow region along the wall. It's one of the reasons why if you want to increase flow, you increase the radius of the tube. And that's because we have so much friction on the walls. But then that brings us back to that figure I showed you before, where we'll look at the dissipation of pressure over the distance and how we get to a low pressure return from a high pressure output. And that's largely because we've dissipated much of that energy of the friction of the fluid movement along the walls of the tube. And if there's time, we'll talk a little bit about the components of blood. But let's start with a figure. And this is nothing more than the 3-box model we talked about last time. And again, it's worth looking at this because this is the major idea of the autonomic nervous system. And what it's showing is this is the central nervous system. This box is the central nervous system. Neuronal cell bodies always found in the central nervous system. This is going to be the cell body of the preganglionic neuron. Doesn't matter whether we're talking sympathetic or parasympathetic, this is the cell body of the preganglionic neuron. Sends a process out, goes into the periphery. That process now is going to innervate postganglionic autonomic neurons in the autonomic ganglia. So this circle is meant to represent the second box that we showed yesterday. This is, in fact, the autonomic ganglia. Preganglionic synapses into postganglionic, and postganglionic now synapses onto the target. And the target is shown here as either smooth muscle, gland, or cardiac muscle. Now we're going to emphasize today not just the muscle, we're going to emphasize that the innervation of the cardiac myocytes will also be the pacemaker cells. And we'll walk our way through who those are in terms of the G protein coupled receptors. This is simply, again, the pharmacology. And what we see with the pharmacology is that always pre to post, whether we're talking about sympathetic or parasympathetic, the pre to post synapse is going to release acetylcholine. This activates ligand-gated ionotropic channels, sets up EPSPs, action potentials, and they propagate through the postganglionic neuron and release neurotransmitter at the target. Now, we're going to see that there's a difference in the neurotransmitter that's released at the target. Norepinephrine is released by the sympathetic branch, whereas acetylcholine is released by the parasympathetic branch. So hold on to that pharmacology. And then this is the anatomy. We had some ice cream cone diagrams yesterday, and this is a prettier picture of that. And it shows more detail, too. This shows up here the cranial nerve 3. This is going to be cranial nerves 7 and 9. Don't worry about those numbers. The only one we're worried about is 10. Because remember, we said cranial nerve 10, the vagus, would exit the brainstem and then send processes throughout the thoracic and abdominal cavities. There's an additional innervation out of the sacral cord. There's some argument about this. But the ones we're interested in are the preganglionic parasympathetic neurons that are going to end up innervating the heart. And so these neuronal cell bodies in the motor nucleus of 10 send a process out, and they now will innervate the postganglionic neuron. So this is a preganglionic neuron innervating a postganglionic neuron in the cardiac ganglion. Remember, in the postganglionic system, in the parasympathetic system rather, that the ganglia are in the walls of the organ. So in this case, we have the cardiac ganglion. It's in the wall of the heart. The sympathetic branch is shown over here. Sympathetic branch, we're located with the cell bodies of the preganglionic neurons between T1 and L3. These neurons extend processes into the periphery, and they synapse onto postganglionic neurons in the sympathetic chain ganglia. And those postganglionic neurons now head out and innervate all manner of things in the head and the abdominal cavity and the and the thoracic cavity. What we're interested in here is the heart. So this preganglionic neuron sends its process out into the sympathetic chain ganglia, synapses onto the postganglionic neuron, and that now innervates the heart. But where we want to go now is to think about what happens at the site of neurotransmitter release onto the target. And that's where we started last time. And we're going to begin here describing the effects of acetylcholine. So we'll take this as describing the effects of acetylcholine on pacemaker cells. In the SA and AV nodes. And we can describe this here. It's a box. This is meant to be a pacemaker cell. We can show the postganglionic parasympathetic neuron here. This is meant to be the postganglionic parasympathetic neuron. The story for transmitter release is going to be the same as we talked about before. Action potentials arrive, borders gated calcium channels open, the calcium comes in, binds synaptotagmin, snare complex, we release neurotransmitter. That neurotransmitter is acetylcholine. But now what we have to think about is the receptor for the acetylcholine. Now, one of the critical ideas in neurobiology is that the variation in the response to a particular neurotransmitter depends upon the receptor. The receptor is the determining factor of what a neurotransmitter is going to do. And so in this particular case, we have an old friend of acetylcholine. But we're going to see it does something very different than what we're used to, because we have here a G protein coupled receptor. 1, 2, 3, 4, 5, 6, 7. This particular G protein coupled receptor, has a name. It's called a muscarinic. Go here, muscarinic receptor. As we get down into the weeds of these G protein coupled receptors, we find that this is only one type of muscarinic receptor. This is said to be M2. We're going to talk about other muscarinic receptors later in the course, and some of those are M1s and some are M4s. That's not a concern, but what I want you to see, though, is that there are variations in the muscarinic receptors. The muscarinic receptors as a class get their name because these these receptors are they're activated by an alkaloid we find in mushrooms. It's called muscarine. And I may have mentioned this yesterday, that if you go hiking around here in the springtime, you'll often see up in the mountains these exquisitely beautiful mushrooms, bright red mushrooms with these sort of beige-like squares on the top of them. That's Amanita muscarina. That's the same kind of mushroom from which we can extract muscarine. It's that alkaloid that will activate these muscarinic receptors. But that's simply where the name comes from. We're not activating these M2 receptors with muscarine in biology any more than we're activating nicotinic receptors with nicotine. In biology, what we're doing is using acetylcholine to bind to the receptor. And from here, the story is pretty familiar. Remember that attached to the cytoplasmic leaflet of the membrane is this so-called heterobifunctional trimeric GTP binding protein. There's some meaning in the translation of this. It's heterotrimeric. Tri means three. There are three proteins. It's hetero. There are three different proteins. And in fact, if we look at it, there's an alpha and a beta and a gamma. So there's three subunits in this GTP binding protein. This is, in fact, the protein that's going to bind GTP. And you may remember from our earlier discussions that when this alpha subunit has covalently attached to it a GDP, this alpha subunit is inactive. And it's matched up with the beta gamma, and all three of those proteins will live happily together. But what will happen is that when acetylcholine binds this muscarinic receptor, someplace in the cytoplasmic side of this, we usually show it near the carboxy terminus, but it's probably a combination of a number of these loops generating a three-dimensional shape into which the alpha subunit can fit. When this binding site for the alpha subunit opens up, the three subunits come in and the alpha subunit nestles into the binding site. And when the alpha subunit nestles into the binding site, this GDP is replaced. There's in fact an enzyme which releases the GDP and replaces it with GTP. It's called a guanyl transferase, and we're just taking the GDP off and putting the GTP on. Now, once the GTP is on the alpha subunit, the beta gamma go their own way. They separate. And the alpha subunit now remains attached to the cytoplasmic side of the membrane with a GTP on it. The next character in the play is this protein that goes across the membrane. Let me put another color in. Green, nope, red. It goes across the membrane like 24 times. But this protein is called adenylyl cyclase. And what adenylyl cyclase does is it takes ATP, adenosine triphosphate, and converts it into cyclic adenosine monophosphate. If you look at the five-membered ring of the sugar, what happens is if this is the five-membered ring of the ribose, 1, 2, 3, 4, and 5. What will happen is that the phosphate up here, there'll be three of them. 1, 2, 3. This is the triphosphate. This is a ribose because down here it has an OH. And out here will be an adenine. So it's adenine triphosphate. Adenosine triphosphate. So now what we're going to do is we're going to use an enzyme called adenylyl cyclase. And adenylyl cyclase is going to take these two phosphates and clip them off. So adenylyl cyclase takes off those two phosphates, and then in exchange it takes this phosphate, which had been attached to the number 3 carbon. Remember, there's an OH down here. So this takes a carbon, oxygen, phosphorus, oxygen, and we're now bound to this oxygen. This oxygen is the phosphate. So we've taken this phosphate, and now we've linked it to the number 3 oxygen down here, and this is now another phospho diester linkage. We've cyclized the molecule of ATP. Seems like a simple thing to do. But once we've cyclized the molecule of ATP and made this into cyclic AMP, this now has a whole new set of characteristics. Is there some confusion here? Can I clear something up? I mean, if you have some confusion, just ask the question. I'll be glad to try to clear it up. Okay, you're doing fine, yes.
I just have one question with the cyclic adenosine monophosphate. That's the CAMP abbreviation, correct? Or it's created after clipping off of those two phosphates?
So you clip the two phosphates off, because we start out with ATP, right? So that ATP tells you there are 3 phosphates. You take 2 of those phosphates, get rid of them. You leave 1 phosphate, and then that phosphate was linked to the number 5 carbon. And now you link that phosphate back around to the oxygen on the number 3 carbon. And this now makes cyclic AMP.
Thank you.
Okay, so that's how it gets formed. Now this molecule of cyclic AMP now has a whole new set of characteristics. And it can now do other things. But let's think first what's going on in here. So what we've done here is we've activated the alpha subunit. We've turned on the adenylyl cyclase. We've increased the concentration of cyclic AMP because we've activated this enzyme to convert ATP into cyclic AMP. This cyclic AMP now, remember, this is a kind of cyclic nucleotide. And remember, one of the major players across the membrane is the HCN channel. And I'll review it for a moment here. If we talk about an HCN channel, it has a channel in the middle, but it's made up of 4 subunits. Each one of these 4 subunits has a binding site on it for CAMP. And we said that the more of these binding sites that are occupied, the greater the probability the channel will open. And if the channel opens, once this channel is open, this now becomes permeable to sodium. Sodium's carrying positive charge. So the greater amount of cyclic AMP we have in here, the more sites we have bound to the HCN channel. The more HCN channels where we have cyclic AMP bound, the more likely it is they're going to open. If they open now, ions are going to flow in, so it brings sodium in. Well, let's go back to the upper panel and remind you what we've talked about with that pacemaker action potential. When we talk about a pacemaker action potential, remember there's this gentle slope, the ramp potential, reaches threshold, goes back down, and then there's the ramp potential. Remember what is producing this depolarization. This is sodium influx through HCN channels. So that positive charge is being delivered to the inside of the cell. It's depolarizing the cell. Now we have to think of a rate idea. I mean, how fast are these channels opening? The faster the HCN channels open, the more sodium is going to come in. The more sodium is going to come in, the greater the depolarization. So we can think about changing this rate of rise. The rate of rise of the ramp potential depends on the number of open HCN channels. So you notice what I've done. What I have done is I have activated this alpha subunit. And I actually have done this badly. I've activated this alpha subunit. And this is going to be G alpha S. We've stimulated this adenylyl cyclase. This is the story I should have told you for the adrenergic system, for norepinephrine. I and I started here, and what I did is I turned it on its head. I wanted to do this for parasympathetics. Maybe I should just start over. Okay, let's start over. Let's begin here with acetylcholine. We can leave the adenylyl cyclase in place. And what we're going to do is we're going to bind acetylcholine to this muscarinic receptor. We're going to have same story, alpha, beta, gamma. It will have a GTP here. But if acetylcholine binds to this receptor, it's now going to allow the alpha subunit to bind to the cytoplasmic side of this protein. And again, the guanyl exchange factor is going to produce a GTP here. But now in this case, what we're going to be doing is we're going to be activating this alpha subunit. But this alpha subunit now is going to go over to the adenylyl cyclase. And because this is G alpha I, because we're working through acetylcholine, the G alpha I is going to inhibit adenylyl cyclase. And by inhibiting the adenylyl cyclase, we're now going to reduce the activity of the HCN channels by reducing the amount of cyclic AMP. So if we reduce the amount of cyclic AMP, we come back to this story. This story still pertains. We still have sodium influx to the HCN channels. We still have the ramp potential. But now, because we have used acetylcholine through the muscarinic receptor to reduce the amount of cyclic nucleotide, cyclic AMP, we now have less of the cyclic nucleotide available, and this ramp potential is going to get more shallow. It's going to get more shallow because we have fewer, let me just do it this way, because we have less cyclic AMP, fewer HCN channels open per unit time. So that means the rate of rise will be slower. And remember what we're trying to do with these ramp potentials. We're trying to reach this magical point here of threshold. It's threshold for the voltage-gated calcium channels. So under parasympathetic drive, where the parasympathetic system is predominant, acetylcholine is released. The acetylcholine binds to the muscarinic receptor, works through G alpha I, slows down the generation of cyclic AMP, so we now have fewer open HCN channels. We have slowed down the heart rate. So now that I've bollocksed that, do you have some questions? Yes?
The receptor for parasympathetic M2 also, or?
The parasympathetic receptor is the muscarinic receptor M2.
And is that the same for sympathetic?
The sympathetic system, now we're going to head to that now. In the sympathetic system, the receptor is going to be a beta-1 adrenergic receptor. And the beta-1 adrenergic receptor is going to bind to norepinephrine. Okay? Yes?
So then G alpha I is specific to the parasympathetic nervous system to slow the heart rate down.
G alpha I is connected to the parasympathetic nervous system. But its connection to the parasympathetic nervous system is through that muscarinic receptor. So the acetylcholine binds the muscarinic receptor. That then activates the heterotrimeric GT binding protein. But that alpha subunit, that particular alpha subunit now, goes over and turns off or slows down the activity of the adenylyl cyclase, leading to a reduction in the concentration of cyclic AMP. Okay? Yes?
The muscarinic receptor, is it unique to the parasympathetic tract?
Is the muscarinic receptor unique to the parasympathetic nervous system? My first, well, let me qualify that. Where we're going to find the muscarinic receptor is, in our story here, primarily in the autonomic nervous system, the parasympathetic branch of the autonomic nervous system. But if you get into the central nervous system, there are muscarinic receptors there too. And so there are muscarinic receptors in the central nervous system. I think they come in all manner of varieties. At least M4 and M5, I think, are in the central nervous system. So that's not part of the autonomics, but it's in the central nervous system. When we're talking about the autonomics, then the muscarinic receptors are confined to the parasympathetic branch. Does that cover it? Okay. Yes?
So just because we talked about both of them, so GS is going to make ramp steeper?
Yes. I will go through that next, because I made a mess of this. Let me go through the sympathetic branch next, and then you'll have the two to compare. Yes?
Does G alpha I turn off the production of camp completely or just slow it down?
G alpha I will turn off the production of CAMP.
So does that mean we won't have any action potentials fired?
So you have less CAMP. So if you have less CAMP, you have less CAMP available to bind to the HCN channels. So you have, because you have less CAMP available, the probability of opening those channels is less. Yes?
Can you please remind me where G alpha I comes from?
So G alpha I, let's do this. G alpha I is inhibitory for adenylyl cyclase. G alpha S is stimulatory for adenylyl cyclase. Then the third one is G alpha Q, and this is stimulatory for phospholipase C. This leads to calcium release from internal stores. We'll run into all of these, but we're not going to be dealing with G alpha Q today. The ones we're going to be talking about today are just G alpha I and G alpha S. And the G alpha I is going to work through M2, and G alpha S is going to work through beta 1. Okay? Yes?
But where does G alpha I come from?
Where does G alpha I come from? G alpha I is among these proteins that the cell generates. And it generates alpha subunits, beta subunits, and gamma subunits. And the G alpha I's are just the alpha subunit. And as far as I know, they will be specific for the beta gammas. So there will be a whole flock of these GTP binding proteins. But what distinguishes them one from another is the alpha subunit. Having said that, I'm sure that the alpha subunit of the GIs and GSs will bind to a different set of beta gammas. Is that? Not sure I'm clearing it up. So you have genes in all of your cells that will generate the production of these subunits, alpha, beta, gamma. And as you generate those, now I don't know where in the process of generating those proteins that they come together. I think they're separate, they're generated separately, They're incorporated into the membrane. Now, they're not transmembrane proteins. That's a detail, but they're not transmembrane proteins. The alpha and beta and gamma subunits are tied into the membrane by fatty acids. So the fatty acids are a part of the protein, and they're linked into the cytoplasmic leaflet of the protein. So they're not transmembrane proteins. But where they're coming from is they're being generated by the genes in your cells. And those genes, because these are not going to be transmembrane proteins, these are going to be made on free ribosomes. And then they're going to be incorporated into the membrane through some enzymatic activity that will add usually a palmitic acid or a myristolyte. So they are one of the myriad of proteins that are in your cells. And they happen to be enriched, particularly in these cardiac myocytes. Okay?
The G alpha I is just a type of alpha subunit.
I'm sorry?
The G alpha I is a type of subunit.
The G alpha I is the alpha subunit. So the terminology, let's go back to this. The G tells you it's a GTP binding protein. The alpha tells you about the alpha subunit. The alpha subunit can be inhibitory, it could be excited or stimulatory, or it could be one of these Q ones that does something different. But the name of the protein is determined by the alpha subunit and whether it's inhibitory or excitatory. Okay? Okay, let me go to the back panel and let me finish the story. And let's take the story, let me deep six this first. Let's take the story we have above. And where the story above was for the parasympathetic branch, this is going to be for the sympathetic branch. And for the sympathetic branch, again, we're going to have a pacemaker cell. This is the axon terminal. This is the axon terminal of the postganglionic. Sympathetic neuron. The neurotransmitter that's in the vesicles of this postganglionic sympathetic neuron is norepinephrine. When the vesicle fuses with the membrane, it releases the neurotransmitter, norepinephrine. So this is going to be norepi. The norepinephrine now is going to bind to a G protein coupled receptor. This G protein coupled receptor here goes across the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. This norepinephrine receptor has a name. It's called a beta-1 receptor. Sometimes you'll see referred to as a beta-1 adrenergic receptor. The norepinephrine binds to the norepinephrine receptor. The story that we have above is pretty much repeated again, but with a slightly different twist. Again, here's our alpha subunit. Here's our beta, here's our gamma. When the beta and gamma are together, the alpha subunit has attached to it GDP, guanosine diphosphate. When the binding of the norepinephrine to the receptor opens on the cytoplasmic side, the binding site for the alpha subunit nestles into the site. The GDP is exchanged for GTP, and now this GTP becomes active. It separates from the beta gamma. And over here is again, I may as well put it in the same color, is again that transmembrane protein. This is adenylyl cyclase. Adenylyl cyclase, again, converts ATP to cyclic AMP. So now what we've done is we've turned on this alpha subunit. Now this alpha subunit is G alpha S. It's stimulatory. It's stimulatory to the adenylyl cyclase. So it increases the activity of the adenylyl cyclase. The function of the adenylyl cyclase is to make cyclic AMP. So through this pathway, we're going to increase the concentration of cyclic AMP. If we increase the concentration of cyclic AMP, what are we going to do to the HCN channels? We have more cyclic nucleotide, and the cyclic nucleotide-gated channels, what's the probability of the channel opening with increased concentrations of cyclic AMP? It's going up. So we come back to this figure. Let's redraw it quickly. So now if this is our ramp potential normally, now let's say we increase cyclic AMP. That leads to an increase in HCN channel opening. That means that this rate of rise of this ramp potential is going to be steeper, it's going to be faster. And what we're trying to think about is how long it takes to get to threshold for the voltage-gated calcium channels. The faster the rate of rise, the more rapidly we reach threshold, the more rapidly we fire one of these pacemaker action potentials. So the monocyclic AMP directly affects the rate of rise of the ramp potential, which then directly affects the heart rate because we increase the rate of these pacemaker potentials. So to round that out, the other side of the story is the cholinergic side, where the parasympathetic branch releases acetylcholine, activates the G alpha I, inhibits adenylyl cyclase. So what we're constantly doing is we're constantly in this yin and yang of how much cyclic A do we have. If we're under sympathetic drive, we have larger amounts of cyclic A, our heart rate goes up. If we're under parasympathetic drive, where we have more acetylcholine being released, we increase the activity of G alpha I, we now slow the heart rate. And that's how the heart rate is determined here. Now, next time what I'll do, and what I hope to get to today, is the sympathetic branch also innervates the contractile muscles. And so it's going to have an effect on the contractile muscles. But I bollocks at trying to do two at one time. So I'll finish that story next time.